Hello, welcome to today's lesson. Today we are going to look at how to answer biology practical questions. We now want to have questions on microscopes. Let's look at questions. So you have these two groups of plant tissue under optical microscope. You have Fig 1A and Fig 1B. The magnification is times 400, each one of them. And we will use these photos micrographs to answer the following questions. Now, question I. Suggest the magnifying power of A, the eyepiece lens, B, objective piece lens, that was used to observe specimen Fig 1A and Fig 1B. These are the questions. Now the answers. The eyepiece lens is 10 times. The objective piece lens is 40 times. The microscope that was used to observe these two was the optical microscope. Eyepiece 10 times. Now, eyepiece of optical microscope have this magnification 4 times, 5 times, 10 times. That's the eyepiece lens. The objective lens of optical microscopes, you can have 10 times or 40 times. These are the magnifications that are used in schools, high schools. You can get magnifications that are more like 100 times. If this had a magnification of times 400, then it can only be that the eyepiece that was used is 10 times and the objective piece that was used is 40 times. Because the magnification of the microscope is eyepiece magnification times objective piece magnification. That's how the magnification is done. So we have eyepiece 10 times multiplied by objective piece 40 times. That's how you come to have 400 times. So this is how you find out the eyepiece lens magnification, objective lens magnification that was used to look at a specimen. This is how it's worked out. The two magnifications give how big the tissue is magnified. The next question is measure the length of cell G. So what is the length of cell G? Now, you are going to use your ruler to measure the length. And when you measure the length, it's 1 centimeter. We can also say it's 10 millimeters. That is the length of cell G. All right? Then question II says determine the real size or width or length of cell G. Now, in working out the size, we have been given the magnification. We also know the length of the cell in the photo. And magnification is length of drawing or length of photo or picture divided by length of the object. That is magnification. The actual length, all right? Real size or actual length. Okay, so if we put these together, then we have this. We have 400 is equal to 10 millimeters divided by x. The x is the real size. And the division will come to give us the real size or length of cell G is 0.025 millimeters. This is how you can find the length, all right, of what you have in the photo. Draw separately cell G and cell H. The length of the drawing should be between 5 centimeters and 6 centimeters. Here is a drawing of cell G, and here is a drawing of cell H. You see the differences? Look at this. The cell walls are very thin in here. Here the cell walls are very thick, and it should show in the drawing. Question V. Which of the tissues are the cells specialized? Fig 1A or 1B? Answer is specimen 1B has specialized tissues because the cell wall is very thick. You see, the cell walls are very thin, and you can see the nucleus inside it and the cytoplasm. So this one is not specialized at all. Very young cells. These are quite grown cells, specialized. Good. Goodbye, and thank you.